Describe the production of mRNA or messenger RNA. Now this is a 5-mark question. You will get a lot of these on your A-level biology exam. Particularly on AQA paper 1, you will get 3 of what I like to call these story questions at the end of the exam paper. And these are the questions where if you do enough revision, you can really pick up all of the marks. So let's go through this one. This is basically asking you to describe the process of transcription, which remember is the first stage of protein synthesis. So it takes place in the nucleus. And the first thing that happens is the hydrogen bonds between the bases in DNA are going to break. Obviously, once those hydrogen bonds are broken, you're going to separate those 2 strands of DNA. The second thing that we need to say is that only one of the DNA strands will act as a template. Now, this is different to when you learn about DNA replication. If you think back to that process, for that description, it was really important to say that both strands act as a template. Whereas here, we've just got one strand of DNA acting as a template to build the messenger RNA. The next thing we're going to talk about is the RNA nucleotides themselves. So the RNA nucleotides form complementary base pairs. And this will follow the base pair rule. Okay, so if you remember the base pair rule, an A base on the DNA will join with a U base on the RNA. Remember, there is no thymine in RNA. Remember, there is no thymine in RNA. In RNA, we replace the thymine with the uracil base. So wherever there's an A on the DNA template strand, it's going to pair with a U on the mRNA nucleotides. If there's a T on the DNA, that will pair with an A in the mRNA. Guanine pairs with cytosine, and obviously cytosine pairs with guanine. So that's how the nucleotides are going to line up following that base pair rule. The next thing we're going to talk about is the enzyme that is needed for the process of transcription. So the enzyme you're going to name is RNA polymerase, and the job of this enzyme is to join RNA nucleotides together. And you can get another mark for telling the examiner what bond that enzyme is responsible for making. So it joins these RNA nucleotides together by making phosphodiester bonds. And that is in a condensation reaction. So it's joining the nucleotides together, adding one to the next by making phosphodiester bonds in a condensation reaction. We're going to continue because there are actually other things that may get you marks for this question. For example, once you've made the messenger RNA, at this stage, we actually call it pre-mRNA because it still needs to be spliced before we get the mature messenger RNA that will be used in translation. So pre-mRNA is spliced to produce mature mRNA. This mRNA will leave the nucleus through a nuclear pore where it goes to the ribosome, where it's used in translation. I'm not going to include that in my answer, only because it's just asking me to describe the production of mRNA. So I'm going to stop at this point, but remember from there, will leave the nucleus, go to the ribosome, and from there we would be describing translation, which is not required for this particular question. I hope that's been useful. Follow me for more advice on tackling these extended response questions on your biology A-level.